Look at a cross-section of a flowing vein, and you'll see millions of red blood cells drifting passively, carried by the current like rafts. But look closer, and you'll spot something else entirely. A solitary cell that ignores the current, grips the vessel wall, and swims upstream. In our last episode, we explored those drifting red blood cells and their tireless oxygen delivery. Today, we're tracking the anomaly, the leukocytes, commonly known as white blood cells. Unlike almost every other cell in your body, leukocytes aren't locked into a fixed tissue. When they detect a chemical alarm, a leukocyte shapeshifts, flattening to squeeze directly through the solid microscopic walls of your blood vessels to enter the surrounding tissue and hunt threats. Far from being passive fluid components, they act as an independent, roaming military force, operating completely autonomously inside of you. To actually see this army, we have to pull it out of the bloodstream. When a lab technician takes a vial of donated whole blood and spins the sample rapidly in a centrifuge, gravity forces the heavier elements to the bottom, splitting the liquid into distinct bands. At the bottom sit the packed red blood cells. At the top is the yellowish plasma. Caught right in the middle is a razor-thin white layer called the buffy coat. That tiny fraction contains every circulating white blood cell. This flowchart shows the buffy coat method used by blood banks. It illustrates whole blood undergoing a heavy spin in a refrigerated centrifuge, physically splitting the donation into packed red cells, plasma, and that concentrated middle buffy coat. Notice the final center step. After a lighter spin to extract platelets, the protocol explicitly requires the buffy coat to be discarded entirely. Blood banks intentionally strip this layer out of donations to protect the patients receiving the blood. Even though it takes up barely a fraction of the test tube, the buffy coat houses the most biologically aggressive and dangerous elements in human blood. White blood cell is actually an umbrella term. Zoom into that discarded layer and you'll find a diverse ecosystem of specialized units, each engineered for a specific type of combat. First are the neutrophils. These are your immediate first responders. The moment a bacterial infection breaches your skin, they swarm the area in massive numbers, sacrificing themselves to neutralize the initial wave. Next are the lymphocytes, which include your T cells and B cells. This is the targeted intelligence division. They memorize the exact chemical signatures of viruses you fought in the past, granting you long-term immunity. Then you have the macrophages. As the heavy-duty cleanup crew, a macrophage physically engulfs dead tissue and live bacteria alike, traps them in a digestive bubble, and permanently destroys them. Human immunity relies entirely on this precise combination. The swarm, the intelligence, and the heavy cleanup work together to form a comprehensive defense network. To find their targets, leukocytes navigate the vast highway system of your veins by tracking microscopic chemical distress signals. Damaged cells release these trails, and the white blood cells follow the scent directly to the breach. The aftermath of their arrival is brutal. That yellowish pus you see forming in an infected cut is a mass grave packed with dead pathogens and the thousands of heroic neutrophils that died destroying them. Because they are so relentlessly aggressive, modern blood banks employ a process called leukodepletion. Donated blood is forced through a dense, microscopic filter. The smooth red cells slip right through, while the spiky white blood cells get snagged and permanently removed. This filtration is mandatory because donor leukocytes are fiercely territorial. If you transfuse active white blood cells into a vulnerable patient, those cells can look around, identify the recipient's entire body as a foreign threat, and launch a massive, coordinated attack. For severely immunocompromised patients, simple filtration isn't even enough. Hospitals expose the blood to intense gamma radiation before transfusion. This shreds any remaining leukocyte DNA, neutralizing their ability to divide and preventing a fatal reaction. The autonomous aggression that keeps you alive against daily infections makes these exact same cells a deadly weapon if they enter the wrong host. Because these frontline fighters die by the millions every day, your body has to constantly replace them. Deep inside the hollow center of your bones, marrow stem cells run a continuous manufacturing line, pumping fresh leukocytes into the bloodstream. Doctors monitor this production line through a white blood cell count. A sudden spike in the number tells a physician your body is actively fighting a hidden infection. A dangerously low count means the factory has stalled, leaving you completely defenseless against even a common cold. Sometimes the manufacturing process itself breaks. In leukemia, the bone marrow suffers a catastrophic failure, turning out millions of mutated, abnormal white blood cells that refuse to die. These broken cells don't protect you. Instead, they rapidly crowd the marrow, physically blocking the production of healthy red cells and platelets. To manage this severe imbalance, modern medicine relies on advanced apheresis machines, spinning the patient's blood to physically extract the suffocating surplus of white cells. Surviving the microscopic world requires a potent military, but true immunological health means maintaining strict, regulated control over that army, because unregulated biological power eventually turns the body against itself. Your bloodstream balances two distinct forces. Passive, drifting red cells carry the oxygen that sustains your tissues, while aggressive, autonomous white cells wage the brutal microscopic wars required to protect it. But what happens when the battlefield itself gives way? When the physical walls of the blood vessel are torn open? That emergency requires a completely different mechanism. Enter the platelets, tiny, hyper-reactive fragments that deploy in seconds to plug the breach and seal the leak. Next time on Blood Explained, we'll dive into the incredible chain reaction of clotting. Subscribe to the channel and join us for Part 4 to see exactly how your body stops itself from bleeding out.